talking about, yes, we're talking about factors that affect how an enzyme would actually perform. And we had talked about how when we either boil something or if we freeze something, then that particular enzyme is outside of its operating temperature, and we said that what would happen to it? Right, it would denature. And denature meant what? Yeah, so in other words, it would unfold out of its natural state, okay? So you want to say it would unfold out of its natural state, because we know the natural state of an enzyme is going to be in that three-dimensional structural form. Okay? Today's 10/18. Okay, I'm going to send the row sheet around. And we said that depending on how far we were out, right? Because we might just get it cold or, warm it up, a little bit beyond its normal. But we said if we weren't too far, we just unfold it. But we said if we boiled it, for example, then we said we could possibly do what? Damage it how? Right, so we've already unfolded it. So we've done what did we say we'd do to the, we said it's in its linear structure, but if we were too far out, what would happen to it? Huh? Absolutely. He said broken down into peptide fragments. So, and we know that once we've gone that far, and then sometimes even once we've unfolded in the linear structure, we may not ever get it to fold back up. But we know that this guy will not be active again. It cannot bind substrate. All right, we said pH, same thing. So in other words, if something is too acidic, meaning that we've got a lot of hydrogen bonds in there, probably covering up things, and if we're too basic, we're throwing in a bunch of these hydroxyl groups, it says, and this is going to interact with those hydrogen bonds that are occurring generally between the R groups, okay? And not that hydrogen bonds are not occurring somewhere else, but mainly they're in between those R groups. It says, because we know that we can damage or alter the three-dimensional structure. So it's not just the hydrogen bonds that we can affect other bonds, like if ionic bonds are occurring between the different amino acids. So it says anytime we're outside of the operating range, whether we're talking about temperature or pH, we end up denaturing the protein. Okay? So the whole idea is we end up denaturing the protein. And remember, D means what? To remove what? Its natural state. Okay? So D means to remove. Okay? So when we complete, remember chapter 5, you're going to have an exam. We've got 2 parts to 5 to do. The first part is the energy related part, and then the last part would deal with the movement of different materials across the cell membrane. All right, so enzymes work in teams. And what we mean by that, since we're not doing the chapter 7 and we're not doing the chapter 8, but still we need to know that enzymes work in teams, and they're usually involved in what we call a metabolic pathway. A metabolic pathway, for example, when we take glucose and as oxygen is infusing into the cells, we're able to break that glucose down into water and carbon dioxide, okay? It says, now, in order for us to get from glucose all the way down to the water and carbon dioxide, it says that we're going to change that molecule glucose into many different substrates and then products before we get to the carbon dioxide and the water. So it says these enzymes, and it's going to take more than one, they're going to be involved in these enzymatically controlled steps that results in the completion of a product. If you were to turn in your books to page 127. Oh, it's marked. I just don't want to lose your page. If you look at page 127, it says we're going to start with glucose. But it says that we're going to put some energy into the system to initiate the splitting of glucose into an intermediate that we're going to call eventually P-gal. And then it says we're going to input at least 2 of those ATP molecules to get that done. So it says anytime you input energy, basically, and we know that energy form was ATP, it says that we're going to end up phosphorylating the molecule. So if you look at glucose, it says after we've put in the 2 ATPs, we end up with a what? 1, right? 3 biphosphoglycerate, okay? So it says this is actually a 1,6. It's because we got a phosphate group that gets phosphorylated on the first carbon, then we end up with 1 on the 6th carbon. And so that's a 1,6. Actually, I think it's a biphosphate. Okay? Now, but then it says eventually we're going to end up with this glyceraldehyde 3-phosphate. If you look in the lighter shaded area, you'll see something that says G3P. That's a glyceraldehyde 3-phosphate. So it says, now, the molecule, or the enzyme that bonded to glucose, it says, is that the same enzyme that's going to bind to the 1,6 biphosphate? Would it do that? Would that be the same one? No. It says, so that's the reason why I'm saying there's a metabolic pathway that's actually involved here. It says where we have a set of enzymatically controlled steps or reactions. So it says as one enzyme binds and produces the product, which is the 1,6 biphosphate, it says now we have to have to that product now becomes a what? A reactant in the next reaction, and then the enzyme that binds that is what's going to create the 2 G3P molecules, which is the glyceraldehyde 3-phosphate. Does that make sense? And then you're going to have an enzyme to bind those in order to eventually get us all the way down to the bottom where we're going to have that pyruvate. Cool beans? So it says each enzyme has a particular task to do, and it says then you're going to have another enzyme to come in to perform the next task. So again, we said, so the product of one reaction always becomes the what of the next reaction? The what? Beautiful. The reactant, what's another word for it? Beautiful. So we started with glucose, and we ended up in that very first, was actually 2 different reactions. They went ahead and did both of them, but we had a 1,6. I think it's, okay, and then there's something else, and I think it's fructose. All right, so but what we're saying is, then to this, we've got to add what? Another enzyme. So this is now the product, but this product becomes, you said, the substrate of the next reaction. Okay, so, and that's what this will say. So it says the product of one reaction always becomes a substrate for the next. Okay, and once we get down to the bottom, we know that that final product that we get to is the substrate of the next reaction group. So we'll start this thing all over again with a different reaction group. So it says this part that's going to take us all the way down to the two pyruvates that you see at the bottom. This whole thing is an enzymatically controlled reaction, but this is only one step out of cellular respiration. This is called glycolysis. Only one step out of cellular respiration. So it says, when we start talking about cellular respiration, we're going to take glucose, okay, and we're going to convert it into carbon dioxide. So it's going to take all of these steps to eventually, and these and some more, to get it all the way down to the carbon dioxide. So it's not just a and a B step, but you can see that there's going to be many steps that are going to be involved in order to get us to that next place. Okay. Maybe fructose biphosphate. I'm missing something. Been a while. All right. So then it says these enzymes, as well as the coenzymes, which are going to be the key guys here. So we said enzymes were actually helping to control the reaction, but we have these other guys. We said the coenzymes help to control the reactions as well, because we said that the coenzymes' job was to transfer two things. What were they? Hydrogen ions and electrons. Good. So it says biochemical reactions in living organisms are essentially energy transfer. So in other words, we're just moving this stuff around. All right. So it says we're going to be doing two things. So we're going to be oxidizing, but we're also going to be reducing. But both of those things always take place at the same time. So this is an oxidation reduction reaction. So oxidation is the loss of electrons. So it says when we start breaking down this guy, it says we're going to lose some electrons, but one of the things with oxidation is usually those electrons are accompanied by hydrogen ions. So it says reduction is the gain of electrons. So we're going to have the coenzymes basically acting as cab drivers who are going to be transporting these hydrogen ions and electrons from one part of the reaction to another part of the reaction. So remember we said coenzymes, which family do they fit into? One more time. Nucleic acids. So they are, even though you hear the word enzyme, a lot of people will get them mixed up and say that they are protein. They are not, okay? They fit into the category of nucleic acids, which means that they have three parts that make them up. What are the three parts? Phosphate group, nitrogenous base, and a who? And a 5-carbon sugar. Okay? So these guys, remind yourself, because you're going to see it again. So here's what I said a minute ago. Oxidation and reduction always take place at the same time, because as one part of the reaction is given up, somebody has to be picking it up, okay? And as one part is gaining, okay, he's going to be gaining, he's going to be losing, okay? So in other words, we're just transferring this stuff about. We're not losing it, okay? So we're just moving it around. So we're going to be breaking some bonds, but we're going to be building some too. Okay? And so again, this is the other statement I made. In living things, we got these hydrogen ions accompanied by the electrons accompanied by hydrogen ions. Okay, so usually you see one, you don't see the other one somewhere close by. Can anybody name me at least one coenzyme? Good. NADP plus. Excellent. Okay. All right. So it says in cellular respiration, it says glucose will lose hydrogen ions and become carbon dioxide. So that is oxidation. So it says glucose will lose its hydrogen ions. So that means electrons are going as well. And become carbon dioxide. We want to call this oxidation because glucose is being oxidized to carbon dioxide. Oxygen then will pick up the hydrogen ions and electrons and become water. Anytime you gain electrons, you are being reduced. So this is reduction. So this is an oxidation reduction reaction. In a little bit, we're going to look at the opposite. We're looking at photosynthesis. Photosynthesis is basically the opposite of cellular respiration, where we're going to see what's going to happen to carbon dioxide. Right, so it's going to do, what's going to happen to it? Good. It's going to gain electrons and hydrogen ions and become glucose. So this is reduction. The water is going to lose the hydrogen ions, which the cab drivers are going to be the ones that kind of help get him over to here. So it's going to lose them and become oxygen. So this is oxidation. So we said oxidation and reduction reactions always take place at the same time. As one part of the reaction is losing, the other part of the reaction is gaining. Okay, so here we said glucose was being oxidized, but oxygen was being reduced because oxygen is going to pick up the hydrogen ions and the electrons. Okay? If oxygen isn't present in cellular respiration, basically what ends up happening is, how many people in here, exercise like heavily or something? What happens to your muscles? Huh? No, they should not atrophy. What? Okay, so fatigue. And where's the fatigue coming from? Beautiful. Build up of lactic acid, okay? And the reason being is, if you are not breathing properly, if you're not getting enough oxygen into the tissues, basically once I start going over the electron transport system, that you have to have somebody to receive the energy spent electrons. And the guy that's responsible for receiving energy spent electrons is, where did it go? this guy. So this guy has to pick up the energy spin electrons from the electron transport system because, and I haven't drawn that, so because we haven't gotten that far. But this guy, if he's not present, so in other words, they said you should be exercising. What term do they use? Aerobically, absolutely. That means in the presence of oxygen, because when you're out, when you're not getting that oxygen in, then that's when you start building lactic acid, and it's because you're not able to get those energy spin electrons out of the system. All right, so it says, so we're going to go on. We'll get to that in a minute. All right, so it says in photosynthesis, we said the hydrogen ions are transferred from who? Water over to carbon dioxide. So that means if we're taking the hydrogen ions and electrons from water and flipping them over to carbon dioxide, that means we're making who? Glucose. So it says carbon dioxide is being what? Reduced, which is what we put down at the bottom. And we're going to form glucose. You can see that. Carbon dioxide is going to be reduced, and we're going to form glucose. We said water is being oxidized, so it's losing these hydrogen ions and electrons, okay? No, wait till we get no. Okay, so we said water's being oxidized, carbon dioxide is being reduced. Okay? Now, the other thing that we want to make sure that we do is make sure we know which of the coenzymes are active during which reactions. Did I go too fast for you? I'm back. I thought I was. Now I'm back. All right. So I'm going to write on the board, and it's going to come up again, but the one that Mr. Ireland gave me earlier was NADP plus. So NADP plus, that's a P and a plus, is going to be the nucleotide coenzyme that it's going to be responsible. This is our cab driver. Let me ask you about this cab driver that I put up here. Is that an empty cab or a full cab? Huh? Correct. That's an empty cab. Correct. So this is an empty cab, and the reason why I say empty because we said its job was to do what? Carry electrons and hydrogen ions, right? So if we're looking at, so this guy, what term would we use for this guy? Out of the terms, two terms that we've been using today, what term would we use for this guy? Would he be oxidized or would he be reduced? Oxidized. He has no hydrogen ions, he has no electrons. Okay? So, if we were to write this, we're looking at the difference. So we said NADP plus is oxidized, but NADPH is reduced, okay? That's a plus sign. Okay? The other ones that we use are for cellular respiration, NAD and FAD, NAD plus and FAD. Okay, those are the oxidized versions. Okay? So we said NAD and FAD, these are going to be the ones that are going to do what? Accept hydrogen ions and electrons during cellular respiration. So NAD plus becomes who? NADH. And FAD, oxidized empty cab driver, becomes who? FADH2. Okay? So this is empty. Bless you. And then the NADP, we said, accepts hydrogen ions during the photosynthesis, passes them over to carbon dioxide in order to form glucose. So NADP plus is the oxidized form. NADPH is the reduced form, okay? Ouch. One person brought me flashcards. One. Mm-hmm. Okay. All right, here we go. The electron transport system. All right, now with the electron transport system, this is a series of membrane-bound carriers. And their job is to pass electrons from one carrier to the next carrier to the next carrier. Carriers, they're protein carriers, and they pass electrons from one carrier down to the next one, down to the next one. Oh boy, here we go. I'm gonna let y'all get this one, and then I'll work on my drawing. I think that's too high. It's kind of short. Okay. I'll try not to be lazy. I'll leave it up there. All right, that's better. All right. So it says every time the electrons are basically transferred from one carrier over to the next carrier, it says that they're going to start losing energy. It says, but the energy within this system will be used to generate ATP. Okay? So the energy that's in this system will be used to generate ATP. I'm going to see if this is the end of my slides. Yeah. Okay, well, here we go. Y'all need it back? Okay. Here we go. So this is the intermembrane space. So in other words, if this is the mitochondria, this is the space between the two membranes. So this is the intermembrane space. And then we know that the part inside of the squiggly line is the matrix. So I'm going to use the mitochondria as my example of the electron transport system. Okay? All right, so it says what we've done is taken a cross-section of that membrane. Okay? So that's what we see blown up. That's just this section of the membrane blown up. Okay? Can everybody see the board? Yes? Yes? Okay. All right. So what happens is this reaction is taking place. I'm going to erase my matrix because it's in the way. And then it says the cab drivers have picked up the hydrogen ions and the electrons that have been lost. So here comes, now mind you, we're going to have multiple of these NADs and the FADs, okay, that are coming in, and they're going to be just coming in, but I'm only going to do one, okay? But you're going to have multiple of these guys coming into the membrane, dropping off hydrogen ions and electrons, okay? So NAD comes in, or NADH comes in to the membrane, and he is oxidized back to who? Right, NAD plus. He drops off hydrogen ions and he releases this electron to the electron transport system, which gets picked up by the carrier, okay? So that electron, when he gets passed from carrier over to the next carrier, over to the next carrier, it's losing energy, but it also gives us energy to start pumping these hydrogen ions into the intermembrane space, okay? And there's a video on this, so just in case you don't get it all written down, okay? So what ends up happening is, because remember we said we got multiple of these guys coming into the membrane, right? So what you end up with is a gradient of hydrogen ions across the membrane. So end up with a ton of these hydrogen ions on the outside or in that intermembrane space, and just a few of those hydrogen ions floating around in here, okay? So when you have more of something on one side of the membrane than you do the other, what wants to happen? So what? Beautiful. So he said wants to come in, wants to bring the system to equilibrium, okay? So there's a guy that allows that to happen, and it's called the ATP synthase complex. Because what we've done is build up an electrochemical gradient across the membrane. We said it's electro, why? Because we got these charges, chemical, why? Because it's elements, okay? So what happens is we're going to call this guy the ATP synthase complex. Oh, Okay. The name tells you what is he going to even end up making? ATP. Okay? Now, you remember at the beginning of this energy thing, we said that there was two different types of energy. What were they? Kinetic and potential. Which one do we have right now? Potential. Because we got all these hydrogen ions on one side of the membrane that aren't moving, okay? But when they start to move, because this guy has a channel inside, it says that these guys will do what? Flow back through the ATP synthase complex. And when they start flowing back through here, what do we have? What kind of energy? Huh? Kinetic energy, right? And that kinetic energy, because they're moving, gives us enough energy to take the ADP, that's not a D, DP plus P, and make who? ATP. So the kinetic energy provided by this system trying to bring itself back to equilibrium gives us enough kinetic energy to synthesize ADP plus P to make ATP. Now, remember we said we got some hydrogen ions coming back in. We said in order for you not to build up lactic acid, we had to have some oxygen. So if we look at the reaction, it says we have who? 6 O2s, right? But we're going to change that from O2, because we're going to have to split them, and we're going to have a half O2 present. Okay, so half O2 means we just got a what? O. Okay? So the O's job, the oxygen's job, is to pick up who? These energy spent electrons. Okay? And we've got some hydrogens floating around in here because they're coming back in to the membrane. So it says I'm going to take a half O2 and bind it with what? How many of these to make water? Right. So you see where water's coming from and why we got to have oxygen? Because it's going to bind with that. And so at the end of that reaction, that's where you see it said at the end we were going to make what? Carbon dioxide and who? And water. Cool beans? All right. So we said this guy comes in, drops off electron, hydrogen ions, they get pumped over, you build up in a gradient. When you get so many, they want to do what? Flow back through. That gives us kinetic energy. Take ADP plus P, form ATP. The end. All right. That's your simplified explanation. Okay? So what? Right, absolutely. And that's how we're being able to join the hydrogen and the oxygen. Correct. All right, at the end of part 1. Thank you for helping me. Now let's go on to part 2 of chapter 5. So it says, now, just like that, we're looking at how are we moving things across the membrane. So this guy also had a channel that allowed us to get that hydrogen ion over there, but it wasn't a free, it doesn't allow them to come back in the opposite direction. And it took energy to get him over there. But we're going to look at exocytosis. We're going to look at 3 forms of endocytosis. We're going to look at pinocytosis, phagocytosis, and cell-mediated endocytosis. We're going to look at diffusion, which is part of this, okay? The hydrogen ions diffused back across the membrane through the channel of the ATP synthase complex, diffusion. Why? Because everything always diffuses from a what? An area of who concentration to what? High to low, absolutely. Osmosis is specifically the movement of who? Water. Absolutely. Shoot, we ain't got to do nothing. Let's go home. That's what I'm talking about. Joking. Okay. Facilitated transport. Facilitated transport said we couldn't just move stuff across the membrane. We had to have somebody that allowed it to happen, okay? So usually there's these proteins in the membrane which are going to help facilitate the movement of these different ions, amino acids, sugars across a membrane. And then we're going to have not just facilitated transport, but active transport, okay? Active transport says we got to have something to help it get across. So we got to have some energy to help it get across. So we're going to look at exocytosis, endocytosis, diffusion, osmosis, facilitated, and active transport. All right, let's look at the membrane first. The membrane is referred to as a fluid and mosaic model. So we say it is fluid and we say it is mosaic. Can anybody tell me why it's fluid? Yes. Beautiful. Has the consistency of olive oil. Absolutely. Okay? And the reason why they know that it has a consistency of olive oil, of course, because they've done some tests. And then they also know that it's fluid. And one of the things that we want to note about that it's fluid is that these proteins that are within the membrane, they're not stationary. Okay, or they don't have to be stationary. So in other words, what we're saying is these proteins can move laterally, diffuse laterally throughout this membrane. Okay? How they found this out? They took two different cells and looked at the membrane. And they labeled the proteins in the membrane so that they would know where they came from. So they took a human, took a mouse, and they spliced both cells and what they found out is that after allowing these guys to sit there, that the proteins that were embedded in the membrane, and I guess I can put this back, although it wouldn't look like this. Okay, I'm just going to put it back just for, but the labeled proteins mixed all up. Okay? So they didn't stay where they were before, but they kind of shifted all around. Cool beans? So it says the membrane is fluid because these proteins are free to diffuse laterally within that bed of phospholipids. All right, let's talk about why is it mosaic. Somebody tell me why it's mosaic. What does the word mosaic mean? And this is just showing you alpha helices. What are these? Beta sheets. Okay, because if you're looking at the inner part, looking at a protein, then this is just showing you what it might look like if you saw the whole thing. And this one also has some helices as well. But anyway. All right, so what does the word mosaic mean? Okay. Huh? Okay, so not any symmetry for real. Okay, so mosaic means good, made of many parts. How many people have ever seen a mosaic glass? Okay, right? Beautiful. All these different parts, make up this one beautiful, and all these different colors of glass, they put it together to make some kind of piece of art. Okay, so made up of many parts. Somebody tell me the parts that make up this guy. So we're going to say, that's what the word mosaic means. So we want to say this is made up of many what? Let's make it right for this guy. Made up of many different macromolecules. You already know this is going to be an essay question on the test. If you miss it, shame on you, because I done already told you. So it's made up of many different macromolecules, and those macromolecules, you should be able to name them. So what do you see? You see what? Good. So specifically, what kind of lipids? Good. Phospholipids. So you need to be able to name these. So phospholipids. Good. Proteins. But specifically, what kind of protein do you just see? Good. We're going to say a glycoprotein. So glycoproteins. And I'll say proteins, you said. And then you said glycolipids, I heard. Go lipids. Do you see carbs by themselves, though? No. It's a carbohydrate attached to a lipid. So that's a glycolipid. I see glycoproteins. What's the yellow things? Good. Cholesterol. What is the function of cholesterol? Good. To help give the... So remember we said it was a consistency of olive oil. This helps give it some stability. So the cholesterol stabilizes the membrane. You should know that. Cholesterol stabilizes the membrane. So it says it's fluid because the proteins that were embedded in the membrane are not stationary, but they are free to diffuse laterally. Okay, part 2 of that is it's also mosaic. Why? Because it's made up of many different macromolecules, and some of the examples are phospholipids, glycoproteins, proteins, glycolipids, and cholesterol. Cool beans. So you ought to be able to answer that, because I'm guaranteeing you're going to see it again. All right. Oh, what the peanut butter and jelly. Okay, sorry about that. All right, exocytosis, first process. You should be able to tell me without him looking at it, what is this? What are we talking about? What are we trying to do? Good, so we're trying to move something outside of the cell. So it says large things like proteins must be what? So if I got to get a protein, if I needed to get this guy, if he was inside the cell, not in the membrane, but this guy inside the cell, and I want to get him out, what do I have to package him in? A vesicle, good. So it said vesicles containing large molecules, okay? It says they fuse with the cell membrane and only the contents are released, because we said earlier that the volume of the cell membrane would increase anytime a membrane package fused with the cell. So talking about your gland cells, so anytime you are secreting the contents that were made inside of a gland to the bloodstream, for example, it says if you're making insulin, how am I going to get it out into the bloodstream? I'm going to put it in a package and I'm going to secrete it. Okay? So here's the cell membrane. That's a bilayer of phospholipids. Here's my membrane with the proteins on the inside and caplui. Okay? I didn't do a good job, but I did okay. Okay? So it says, but the volume of the cell membrane or plasma membrane just what? Increased. Only the contents, the proteins that were in solution, were released outside of the cell. Oh, yes, sir. Yes. The cell membrane. Yes, sir. So you should be ready to test on this on Tuesday. Yep. By Tuesday. By Tuesday, that test should be taken. All right, and then this is just showing you more, okay, showing you the intracellular vesicle going out, fusing with the membrane, and now that blue part, which was the vesicle, is showing you that the membrane volume should be increasing because that becomes a part of that membrane. It says, well, what do we use this for? Antibodies, for example, neurotransmitters. If we're looking at the end of a neuron, and in order to get that neurotransmitter outside of the cell, usually it's packaged in a vesicle, okay, and then secreted outside of the cell, okay, and then you've got either another neuron or a muscle waiting with receptors on it to receive those neurotransmitters. It's online. All right. Endocytosis. With endocytosis, we're saying we're going to bring something from the outside to the inside, but depending on whether it's a whole cell or whether it's something in solution is going to determine what form of endocytosis we use. And then sometimes we're even more specific, so we'll use another type of endocytosis. So two main types that we want to talk about are pinocytosis and phagocytosis. Pinocytosis, we know, basically translates as cell drinking. So when you have things or molecules that are in solution, this is how we're going to bring them in. But if we've got a whole cell, like a bacteria cell that needs to be engulfed, that's going to be a cell eating, okay? Because we're going to engulf the whole cell. Nothing is in solution, okay? So phagocytosis. So pinocytosis, again, translates as cell drinking, and phagocytosis would translate as cell eating. Remember, whole cell parts in phagocytosis. Whole cell parts. Okay, so it says in order for us to make this vesicle, or to get the contents that are outside of the cell. So here's my cell, okay, and here's the membrane, but it says in order to get the stuff in, it says the membrane has to do what? It has to invaginate, okay? So that's an invagination of the membrane. And it says then whatever's on the outside can be brought and packaged in this vesicle. Okay, so we actually invaginate the membrane. So you see here the invagination of the membrane. So we've got the contents of things that are in solution on the outside of the cell, and the cell membrane will actually invaginate in order to bring those particles inside the cell. Okay, so it says anytime we want to bring those things that are in solution, again, inside the cell. So again, we said this translates as what? Cell drinking. Cell drinking. One word that you might want to go back and put on the phagocytosis. Have we done phagocytosis yet? Oh, okay. It's coming. Thank you. Okay, so here we're actually forming a phagosome. So remember we said this is something that's not in solution. We're talking about a whole cell part. Okay, for example, a whole bacteria cell. And in order to get that bacteria cell inside of the neutrophil, for example, the neutrophils, which are normally the first defense guys that we have at a wound site. So if you get cut on your finger, these are the first guys that go out and try to get rid of all that bacteria that you're letting inside of your body. Okay, so we said the phagosome is the package that's actually being created. So we're talking about large things. If you get cut, if you've got a wound, the old cells or broken cells, it says this is how we're going to get rid of those cells. Okay, so at a wound site. You also have some cells, say for instance, I don't know if any of you have ever stepped on a hoe or a shovel and got popped in the forehead and had that big hematoma. Well, there's cells that actually go in and they eat away all that blood that's trapped underneath your skin. You don't have to poke a hole in it, but there's cells that actually go in and clean all that up. Yes, sir.
How fast do these processes take place?
Pretty fast. If you've ever, there's like videos out there that show you how quickly, say for instance, because like your body has chemicals that signal, hey, we need neutrophils like right here now, So yeah, a lot of that. Think about how quickly a wound heals on you, especially if you don't have anything like diabetes or whatever. It's pretty fast. How fast you clot, to seal that, So everything's pretty fast. But if we've got things wrong, then of course it's going to take it a little bit longer. In seconds, I couldn't tell you, or even in minutes, I couldn't tell you, but it's pretty fast. We're pretty neat. Did everybody get all this? Okay. Okay, now this is the one. The only thing that you need to remember about cell-mediated or receptor-mediated endocytosis, you might even see cell-mediated endocytosis, is the fact that you got to have a receptor. And notice that there are green guys out there and there are these blue-red guys out there. But cell-mediated is what? What would you say about it? It's what? Specific. It's only going to recognize a certain molecule that's outside of the cell and bring it in. So it says it's going to be very specific, a mechanism in which specific molecules are ingested into the cell. So we're not going to just bring any and everything in. That molecule, because remember we said proteins have a what? A particular active site. So it's only going to bind certain things. Okay. So that's the only other thing that you want to make sure you always remember about this guy is that there's a receptor involved and that it's specific. That's all you need to know for the test.
That's like insulin?
Correct. Absolutely. All the hormones, are recognized specifically and brought into the cell. That's the reason why some tissues don't respond is because they don't actually have the receptor out there that allows it to be brought in. That is, yes, sir?
What does it look like? Like if you were to look at it with an electron microscope, it doesn't really look like.
No. No.
What does it look? It just.
You'd have to look. Okay, look, say for instance, like the picture I gave you before, the protein parts of it is going to look like that.
But it only recognizes it because it only binds to that.
Mm-hmm. All righty. Diffusion. And I got a few more minutes, not much. All right, diffusion we know is a process that's due to the constant random motion of matter. So in other words, everything is moving, okay? Even the particles in your table, even though they're locked into position, they're still vibrating. So it says the random motion of matter in which molecules would move from an area of high concentration to low concentration. Somebody burped in the back of the room. Okay, it's not going to stay right there by their mouth, but it's going to do what? It's going to diffuse through the entire room. Now, luckily, the folk in the front, if it happened in the back, hopefully won't have to smell that. You know what I'm saying? Okay? Especially if they had a lot of garlic and some of those processed meats like, I don't know, whatever you get at the deli. You know what I'm saying? But anyway, because what's going to happen is the good air is going to what? Jump in there, and then the bad air is jumping out there, okay, until we bring the system to what? Equilibrium. okay? But we're always moving from an area of high concentration to low concentration. But in diffusion, we're always talking about what? Everybody. If we're saying diffusion, we're talking about everything. But when we talk about osmosis, we're specifically talking about what? Movement of water, okay? If we're talking about dialysis, what are we talking about? No, not blood. We're cleaning the blood, but we're moving what? The particles that were in the bloodstream. Make sense? Okay? So anytime you talk about dialysis, you're talking about the ions and other things like that, okay? The chemicals. Anytime you're talking about osmosis, we know it's water. Diffusion is everything. Water and salt and whatever else. Cool beans? All right, so it says molecules follow what we call a concentration gradient. So they follow what's called a concentration gradient, okay? The gradient just means what? A difference, a change, okay? And then it says it follows it based on what? Whatever's naturally due to the motion. We always move from what? High to low. That's natural. High to low. Unnatural is what? Low to high, okay? Now, we want to talk about gases and fat-soluble molecules. Why? It says because gases can do what? They can fit through here. Okay, gases and fat-soluble things. So in other words, if you're outside and you're playing in the pesticides, okay, then what could happen? Absolutely. They're going straight through the cell membrane and they can affect your DNA. Okay? So anything that's fat soluble, you need to have on what? For example, anything poisonous like that, you would have on what? Some gloves. That's the reason why they tell you to put on some freaking gloves, because it's going to go into the cell. Here's your DNA and probably alter your DNA. Okay? So that's the reason why fat. So these are the two things that can easily cross that membrane. We know those other guys that are charged, okay? Because we know that a charged guy coming up just to this, is he going to want to go through there, a charged guy? No. Why? Because this is what? What do we call this part? Polar. He likes that part, but what about in here? What happens when we got in here? Non-polar, right? And they don't like charged guys. Okay? All right, so the rate of diffusion is going to depend on three things. I'm going to give you one. Temperature, give me two more. Good. Molecular weight and the concentration gradient. Okay. So we know temperature, the faster it is, the hotter it is, the faster it's going to move, right? Molecular weight, the bigger the molecule, slower, right? Or the smaller it is, we know the faster it'll move. Okay? But then with the concentration gradient, this is a little bit different. It says, if I've got two solutions, and I'll stop after this, but if I've got two solutions, so here's beaker one, here's beaker number two. And it says I'm going to layer two solutions in both beaker. I'm going to put a 90% sodium chloride solution here, and on top of that 90% sodium chloride solution, I'm going to put a 20% sodium chloride solution. Over here, I'm going to put a 40% sodium chloride solution, and I'm going to put a 20% sodium chloride solution. Which one's going to come to equilibrium first? How? Why? Okay, which one's moving the fastest? He says this. How many people say this one? Okay, how many people say this one? Okay. How many people say they don't know? This one is going to move faster. Why? Because you got a bigger difference. Because the bigger the difference in the chambers, you got a faster urgency to bring the solutions to yes.
It's almost like more pressure.
Right.
More pressure.
Right. And this one, they're pretty close. So, you know, they're going to get there, but it ain't no big rush to bring that system to equilibrium because they're so close. Okay, so we want, yes, sir. And I'm done. So we only got 6 slides left. Make sure you read over that information. The test will start probably on Thursday. Not in here, but it'll start on Thursday.